Well,
कॉर्नफ्लेक्स दे दो थोड़ा अच्छा है कैंडी की थी Very good evening, class. I hope you're all doing fine. We're having this cyclone. Because of that, we're having continuous rain in Delhi today. I hope at your place also it was not worse and everything is fine because of that. So even the 2020 was like that. We had a lot of things side by side Corona, and in this 2021 things are the same somehow. Sorry to start like this today, but that's the reality. Never mind. Um, so we wait for 3 more minutes, and then we start with the cell culture part today. So I mean, at the moment, you might be some students of advanced level, some of you might be of mediocre, or some of the basics level. So these presentations, these workshop is actually created for the all the students who want to get the basic knowledge, whether it's a electrophoresis, whether it's DNA microarray, whether it's a recombinant DNA technology, whether it's microscopy, whether it's facts analysis or bioinformatics. So I try to brought all aspects of the techniques. Your I hope I was creating yesterday night syllabus for agriculture biotechnology and as well as for the microbiology for the MSc courses that we want to initiate. So at the moment because when you are creating a syllabus for one university, so we need to discuss first what other the universities are going through, what other things that is happening in the major universities. So what I discovered that instrumentation and techniques, that was the most common subject in most of the courses. So in that sense, if you have this certificate of MBBT, that will be very value added. And not only that, that you are here for, at the moment there are 30 students, in for registration there are 80. So most of the students I have seen, they come just for the sake of um, for the certificate. But nevertheless, they some might also see these videos later, they might not have time. But please, it's nice if you all could join at the same time, so that we have a good session of students, good Q&A is happening, and I can deliver much information, good information to all of you. So I hope just 1 minute more. Meantime, I prepare the slides. Okay. So today we will focus first of all the basic microscope that you see in your 12th class, um, that we handle the objective lens, eyepiece, holding stand, our sample where we put that. So based on that, we go to advanced microscopy, like your confocal microscopy, um, scanning electron microscopy, transmission electron microscopy. I wanted to also input some um, this there's a Crispo, forgot the name, it's Crispo something. It's latestly being used, highly used in the research R&D world. So let's start. So let's have very basics. This is just class 12 studies that we're gonna do today. Microscope basics. So microscope, it's actually is helpful. In earlier time, the Leeuwenhoek, he was the one who discovered this microscope. He was traveling. He was a trader and was traveling around the European shores in order to buy silk trades, good clothes, and he has a habit of like to check the quality of his clothes. So he made one hobby to see some some bacteria, protozoa, viruses, just to for his own uh, hobby, for his own interest. And he saw many things, and he called as animalcules at that time, and which was also published very nice papers. So that was the one interest. And recently I was going through, I don't know if you are on the LinkedIn, I was going through the post of Nobel Prize winners. So you should also follow them. They are quite inspirational and very nice. Those whoever is getting a birthday or something was has been identified, they are coming under their post. Noble law, no. Hmm? Why is it not coming? It was just coming now. I lost it. Because from Microscope, it reminds me of his quotes. Uh, I just wanted to share with all of you. Yeah, this one. Just read this one. I hope you can read this. Um, Age 12, Randy Shackman was determined to save money from odd jobs to buy his first professional microscope. But he couldn't just reach his goals as of $100 as his mother kept borrowing money from his piggy bank. Yeah, so he was working hard in order to save some money to buy a microscope. And on Saturday, I become so upset that after mowing a neighbor's lawn, so in USA, what happened? All the kids in the younger age, they are working in order to. Their parents told them to earn some money so that they could understand how this world works. So mowing a neighbor's lawn, and I bicycled to the police station. I announced to the desk officer that I wanted to run away from home because my mother took my money. I couldn't use it to buy a microscope. So he went to the policeman and asked there that, okay, my mother is taking money from my piggy bank. I can't buy my microscope. So please help me. I want to get away from my house. So whatever he did in this desperate measures, His parents picked him up from the police station and bought the microscope on the way home. And the Shackman old microscope is now displayed in the Nobel Prize Museum. So he's the person, uh, Randy Shackman. So these are the small stories that he's an American biologist, very famous, very famous one. At the University of California, he's at the moment, and editor in chief of PNS, an annual review of cells and developmental biology. He's still alive, age 72. That's good. And he worked on the DNA replication, DNA resolution and replication. So, I mean, all the stories. I would like to share this with you, this website, because don't confine your studies. What are you studying with me, or in your college, or during your college time? Just this broaden up our thinking that this world is too small. We never know where we are, and anything could happen to us. So be inspired. So that was a small story. Before we start with the microscope, what our microscopes are. So microscopes, they magnify objects and they make objects look bigger. Things we can't see from our naked eye, we can see with the help of microscope. Whether you're doing a cell culture or you're doing wanted to see cell phenotype or want to see if there any infection, wanted to count cell number or look for diagnostic analysis of some diseases. In every field, microscope is very important, quite crucial. So this helps scientists to study objects and living things too small to see with the naked eye. So they always carry a microscope with one hand holding the arm and one hand under the base. So you put one hand here and at the bottom. This is the very basics. In the research labs, we don't use this, but I wanted to show you just to take your old time sake, your 12th class, when your biologist teacher is taking to the biology lab. Students, let's see some slides. Bryophyta, pteridophytes, mitosis, meiosis, or like this. So we used to see this in the class 12th under these typical microscopes. So here we had this ocular lens on the top, body tube, nose piece, objective, stage clips, diaphragm at the bottom, light coming from the bottom, the source. Then there's the two adjustments, coarse adjustments and fine adjustment. That's the stage, an arm, yeah. So all together make your microscope. And this objective could be of three types: 20, 10x, 20x, 40x, or even higher. So there are three types of microscope: simple, which has only one lens; compound, two sets of lenses which could magnify up to 100 to 200 times; and there's electron, which even bigger. And our focus later on for this session of microscopy will be mainly based on the electron microscopy. So basically, we use them in this electron microscope. We don't use any lenses. But with the use of electrons, we see very clear and a large image that is not possible with these lens microscope. And we see very clearly your mitochondria, whether it is any other cell organelle, nucleus, we see very clearly in them, very much. So parts of microscope. So ocular lens, eyepiece. So lens of the microscope that looks through. That the ones at the top. Then the coarse adjustment to bring the object in the focus. Then the fine adjustment to make the finer details more clear. And parts of microscope. There is an arm to hold it to support the body tube. Then the nose piece on the top. Objective lenses could be of 40x, 20x, or 10x. And the lower scanner sometimes, which we used to have in our school, 3x or 4x only. And the middle objective lenses are almost 10 to 20x. So there are three powers possible. That is your low power, middle power, and higher power. Lower ranging from 3 to 4, middle from 10 to 20, higher from 40. Then at the bottom is the stage, and you put your slide is known as clip, and the diagram which controls the light entering coming into it, into the field of view. So each part has been described on a skeleton way. That this is the quite to make it clear. Now, light source. Mostly we use mirrors at the bottom, but sometimes we also use electronic light. Base to support it. That's it. So I will go to the right now one by one in the chat box. Keep telling me what is what. So we start from the right. This one is, what is this? This one is your objective ocular lens, eyepiece. This one is your body tube. Then nose piece. Then objective. Then these are your clips, diaphragm, light source, from here, arm, stage clip, coarse adjustment, fine adjustment, then the base. So we always carry one hand on this and other at the bottom if you're in the lab. So this is the normal, in the normal microscope that we see the field of view, that is the area that we see in this circle through the eyepiece. Oops. If E is written like this, so we will see the opposite and mirror view of it. Always. Opposite and mirror view. So if E is written here like this, it will be like this then. So comparing powers of magnifications. So we can see better details with the higher powers or magnifications, but we cannot as much as the image is there. So there is a 7x this image, and this is your 10x. So same way, if we are seeing some cancerous tetranoma cell in the skin cells. So when we enlarge them, so we can see each particles in higher magnifications in a very fine manner. So like bulb of 1, 2, 3, 4, 5, 6, 7, 8. So same we can see better in this. So to check that what is your power, it's written on there in order to read them. So it's like it's written 10x here. No, this is 40x phase 2. So calculate the power of magnification, multiply the power of ocular lens and the power of the objective. So when somebody asks you to what is the magnification of this particular microscope, what you do, whatever the ocular lens power is and the power of your objective lens, then you multiply them, you can come to the answer. So let's calculate. To find the power of the lens, it is found on the side of the lens. So magnification power of lens is always identified by the label of x, 10x, 1000x. So you multiply with the eyepiece by the power of your objective lens. For example, eyepiece power is 10x, objective is 100x, right? Eyepiece is 10x, objective lens is 100x. If it is eyepiece is 10x, then objective is 50x. So in this time, this will be 1000x. And this will be 500x. 10x is 400x. So appearance of the specimen. So objects appears upside down or backward in them. So if it is just like this, as we have discussed, we will see images like this always. So always upside down. This is if we have in the lab, then we can do it, but not possible now. So this is not for us. And yeah, and if you want to mount a slide, so what you have to do, you put in the center a water mark, a drop of water to clean the slide, and after cleaning the slide you put your sample, and then you take your cover slip and make it to 45 degrees Celsius angle, then slowly cover your cells with it. You can use tweezers to lay specimens on the drop of water or something, and then you will have your sample, this E sample covering the slip. Just take care, no air bubbles. No air bubble should be there, because if you see air bubbles, if you want to take some images or later, these air bubbles create really messy images, and it's not. In any case, whether you're doing your electron microscopy or doing inverted microscopy, polarizing microscopy, DEC, any kind of microscopy, just do take care while making your samples. No air bubbles. Air bubbles should not be there, because when you have them, like you have this very nice, one cell structure is there, and these air bubbles will come and will cover your structure like this or like this. And if you want to save this image for your like presentation or publication, these air bubbles look horrible. It's like somebody's wants to take a picture of herself or himself. You put your some kind of odd things in front of them or over the face. So that's how these air bubbles are awkward. So just take care, no air bubbles. So this is how I have just discussed this. You make it 45 degrees Celsius, put it slowly, lower it down, then see your images. Mutant genes, thank you very much. So that's some funny story at the end. That is, this is a gene. Yeah, this is gene that we are wearing, and it has 3 pairs. not two pairs, two leg pairs, one leg pair, sorry. It has three legs, and it's a mutant. And the scientist who is seeing it is saying that, oh, I found a mutant genes. I mean, it's genes actually, Genes, but some sarcasm is also good for health. Never mind. So we discard the annotations. Microscope Basics. Now we go to advanced advanced microscopy. Though it will be more subjective, it's more about how the principle is and how does it work and what are the things that are made up of. So this will be quite theoretical part, not any virtual lab from now onwards in this part. So be patient with me. Yeah. So as I said initially when I started with the microscopy today, so Leeuwenhoek was the one who initiated because of his own interest. So this is the replica of Leeuwenhoek. At that time, microscope was constructed in this way. So don't ask me how he used to use it. Even I don't know, but I would be happy to know it. Or how this equipment was used as a microscope. I like this image, so that's why I tried to put it here, but it's still mysterious to me. Maybe he put his sample here. And what is the role of this? Sorry, but this is they have this in the Texas University of Texas in the medical library, in the medical branch there. Yeah, this we can say yes. This is a good microscope where we can see some ocular lens, an objective lens eyepiece, having this mirror down, and under this wooden case, beautiful wooden case, um, this golden brass covered cylinders outsides. It seems it's working as a microscope. This seems so far. This is the replica of Culpeper tripod microscope, which was built in 1725 by Edmund Culpeper and the collection of Moody Medical Library. And this is the Marshall microscope by John Marshall from 1663 to 1725. So these are the very classics where they did really good researches. They're still being held on. So that was something about old part. Now let's come to the current and upcoming future about microscopy. So I've divided these types of microscopy into two parts. That is your light microscopy and electron microscopy. Yeah. And under the light microscopy, we will discuss 7 types of microscopy: bright field, dark field, phase contrast, polarizing, DIC, epifluorescence, and confocal laser scanning. Confocal laser scanning or confocal microscopy, you can also say. Then comes electron microscopy, under which you have scanning and transmission microscopy. And under electron, but most likely currently that is used in the day-to-day life in the lab. It's a bright field, an epifluorescence, and confocal. Most likely they are or sometimes also dark field. on like daily basis. So just go through them, we have to be taken care of them. These one, the phase contrast, polarizing, DIC, so they are being used in the special labs where they need of special, they're working on advanced microbiology or working on the advanced ecological paths. So it depends actually. If you're being a part of normal molecular biology lab or biochemistry lab, so these microscopies are highly advancedly used. And once you have identified one structure with the help of these microscopy, then you want to extend your interest to even higher magnification, then you go with the electron microscopy with two subtypes, that is scanning and transmission. Both have different principles, but most likely at the moment, TEM is called transmission electron microscopy. This is most people go for. So these advanced microscopies currently, these electron microscopy in India, they are present not in most of the universities, hardly, maybe in ISC Bangalore or ICERS, maybe they will have, in IITs they might have, but not most of the research institutes, not big institutes. Then we will discuss about specimen preparation in this part, that is your light microscopy, how we prepare a specimen for that, for SEM and for TEM. So that will be the whole content for this microscopy chapter. So as we have discussed, they have been using for the long term for various biological studies and general techniques. And broadly, we have divided our microscope into two parts. One is light and electron. So for the electron microscopy, there is separate component, separate dark room is closed, where your microscopy part is handled over the hydraulics, so that the samples, when you're checking them, they should not, your desk should not move left or right. So that's why they keep on the hydraulics also, because of that reason. So let's start with the light microscopy. First part, that is your bright field. I mean, light microscopy, as it says, the source for working to see your things, we are using light in them. And for the electron microscopy, as the name suggests, electron, we are using source electron for making it done. So the most common general use microscopes. So bright field microscopes are named because the microscopic field is bright, while the object being viewed is dark. So in the bright field, so we have two types of within the light microscopy, bright field and dark field. So in the bright field is the one, and it's a highly used, the one that we're seeing in the right, that's also used in my most of the time in my cell culture, where we just sit. Not this one actually, basically I have used the inverted one. I will show you in upcoming slides. So they have very simple design, and light is directed at specimens. As you top the image from the bottom, you give your image from the top to, from bottom to top. So stained specimen shows excellent contrast, very high numerical aperture, good resolution, bright background for dark specimens, and the light source that we use is halogen or tungsten light source. So this is a binocular bright field microscope with movable stage. Stage could be moved. Dioptric adjustments, condenser, iris diaphragm, and built-in light source. And these are called as clinical research student microscopes. Some of the bright field images. So we will go through one microscope. We'll see a little bit detail about them. What is the basic specific characteristics are, what does they show. Then we see what kind of cells we see under them. So these are the images that we see under bright field images microscope. This one is your stained blood cells in peripheral blood smear. This smear of cells, like you take your blood from your body and you stain some blood cells, and here we can see some like this one. And this is the gut tissue from our intestine, and it's having containing ciliated parasites. So these ciliated means the longer. So these this one. So in the gut, in the section, we can see these parasites. And this is a flagellate trichomonas, moving one, looking like that. This one I had in the bright field, it's a modification version, inverted microscope. This is we have used the earlier one. It's hardly available actually in the lab. It's not being used. This one is being highly used in the labs, in which we put our cell well plate, like it is a 6 cell well plate or 12 cell well plate, or even a slide. We just put it down there and we see things from this side. And our objective lens are at the bottom here, your 10x, 20x, 40x, and allowing of cells in flask, well plates or other deep containers that do not fit between the objectives and stage of standard bright field microscopes. So we have inverted microscopes, positions of light source, objective is inverted, light source is above specimen and objective lenses are located beneath the stage here. Now comes the second type, that is your dark field microscopes. So they create a dark background first of all to view our small unstained objects. So in the bright field, it's a bright, everything is in the light, but in this dark field, the difference is mainly that your background is dark, so that everything, whether it's a motile bacteria, which were difficult to see in the bright field, we can see them in this case. And the central light that is coming is blocked so that only the oblique light strike the specimens. So mainly what we are seeing is our specimens more clear manner rather than like in the bright field. And the scattering light rays then enter the objective to form the image. So this method was created in 19th century. It's used for bright specimens and dark background. Light not scattered by the specimens bypasses the objective, therefore making the field dark. Can see very small objects, but resolution is variable. And it's a high contrast, good for unstained live motile specimens. So dark field images. So as we can see in the background, it's all dark, and the things that we want to look in the both images we can see very clear and shiny. So that's the main advantage. If we see from the light microscopy, here everything is being light, and that's mostly we use in the lab. This is in the advanced cases, somebody is looking really for better resolution image for publication, or for the better research that you're looking for, they can go for this microscope. This is the alga hydrodictane reticulatum, viewed with dark field microscopy. And this is the Leptospira spirochete, viewed with the dark field microscopy. Uh, Then comes the third one, phase contrast, which converts differences in refractive index in a specimen to differences in image brightness. The central portion of the light source is blocked, creating a ring of light from the condenser that illuminates the specimens. Then light waves refracted by the specimens are allowed by a phase retardation plate, increasing the difference in wavelength between refracted and unrefracted rays. So when the refracted and unrefracted waves are focused, they produce an interference due to the difference in the wavelengths, and this is seen as differences in the brightness in the specimen. This is our phase objectives, various phase contrast objectives, and we have this nice annular ring also at the bottom. And this is the technology from the 1940s, and they provide much better than your bright field and dark field, your high contrast and good resolution, good for your bacteria, flagella, cilia, organelles such as mitochondria, and for unstained or live mounts. So let's see some images from the phase contrast image. This is the unstained squamous epithelial cells, then observed with the phase contrast microscopy, and this one is that we will discuss DIC later. So we can see the contrast is the difference, the contrast of the image. You know, it's more better, the things that are present inside the cells, the organelles. This is the nucleus, bright nucleus, shining nucleus. Both are somehow attached to each other, and some of the maybe organelles or debris, I don't know what are these, but still it's not that good image as it should be, because we cannot be sure what are these small particles that we are seeing. We cannot define them. Then comes your fourth one, that is your polarization microscopy, which detects specimens that are birefringent, have the characteristics of double refraction, and the velocity of the light refracted by the substance is not the same in all directions. The specimens is placed between two polarizers crossed at 90 degrees Celsius to each other. So this is your polarizer, and you put your specimen here, and you have another polarizer on the top, and in between is our specimen, our sample. And then we check our image. It's quite good for the molecular structure, crystal minerals. It can be quantitative also. So some images from polarizing microscope images. So here we can see it's mainly used for the minerals that are found in the this one is from the Martian meteoroid, like from the space, and we found this to be 0.3 millimeter depth and viewed under this microscopy, and we can see very clear crystals of that meteoroid. On the top right, this is the renal epithelial cells containing fat globules, and at the bottom we are seeing some of the cells, in the same cells in the higher magnification, and this cross is known as, this cross that we are seeing, Maltese cross, this fat one. So these are some advantages of using polarizing microscopy. Then comes your fifth one, that is Epi Fluorescence Microscopy. So this one is used for UV light source. In this, the source of light is UV light, and we have barrier filters and dichroic prism. So this is something we have added some new stuff to this Epi Fluorescence Microscopy. So this microscopy allows the detection of molecules and ions within the cell. So fluorescent dye that we have added into our cell, they absorb short wavelength of light and emit longer wavelengths. And barrier filters and dichroic prism select the excitation wavelength that strike the specimen and exclude the excitation wavelength from the detector. So allowing only emitted light to reach the detector. So whatever the light that we have added, mainly the one which have strike to our cells, they will be going in and not the other. So light source that we use here is UV light as a mercury or xenon arc lamp. We get really high contrast, high resolution images. Special fluorescent dyes used to locate molecules in specimen. Black background, bright stained specimens. No condensers are required. Multiple fluorescence probes available. And detect small quantities molecules, can use antibody staining technique. Okay, then Epi Fluorescent images. The locations of specific molecules can be identified using fluorescent probes. So this is the immunofluorescence technique with the antibody. We have a probe our sample with a specific antibody which will glow in the dark, such as fluorescein. So in this case, I think we have used fluorescein here also, which is green in color, yeah, fluorescein isothionate. So this is tetrahymena, yeah, tetrahymena that we are using as a protozoan, which emits green light after we have added into our epifluorescent image. So here we can see very nice lining coming up, like straight. So which might be not, we can't see this way of structure in the normal microscope. So this was only, first we probe our cells with fluorescein isothenate, which emits green light, and we stain with the anti-basal body antibody. And in the right case, we have stained with the antibody against cell membrane. So this was done for the body, to see the body, and this is to check the cell membrane surface antigen. So we can compare in the both things that how are looking for, you know. So this is the body, basal body, you know, and this one is for the cell membrane. Both images are different, quite different actually. So cell membrane is actually having these coming up, these, you can nicely see this web-like structure, and the body seems like to have this curve here, very nice curve, small dots. And this image that you're seeing of the same thing in the bright field microscope. So this is how we see, because we can't predict these two different structures until and unless we have that this microscope. Because of this microscope, you're able to predict the structures like this. So that's the things got advanced from your previous microscopy to this much advanced epifluorescent microscope. This is another image, very nicely done, showing the metaphase state in the lung cell. 3 fluorescent probes are there, specific for DNA, keratin and tubulin. And this is the blue one, is the metaphase chromosome, and the red one is the keratin filaments, and the yellow is the spindle apparatus. Like we have this 2 microtubules and spindle fibers coming up here. So this is how the beautiful the microscopy can make our cell biology. So this was not possible until in last 40 or 50 years to see the cell mitosis in this way. Like this. So we can see the chromosomes separately, we can see our spindle fiber separately, and we can see your keratin filaments, your microtubules, all these outside uh, in three separate things in one single image. Wish I could put this in my room as a one picture. So that was the thing about microscopy. So from tomorrow we will start with DIC, differential interference contrast microscopy. We can make our lecture here today. I can see if you have any questions. Okay. So I will share these slides with you, bache. The DIC one tomorrow, all these one microscopy until we finish these slides and continue from tomorrow. Yeah. If you have any questions, because this was very straightforward, we are going one microscopy and seeing its application by seeing their images and seeing differences between each microscopy, how the image looks like, and you can choose if you're lucky, if you're having a very rich lab, because labs have always lack of money, and if you are having a rich lab, then you can choose any microscope you want to buy from Lycia, big company, and then go for it. Okay, class, then thank you very much. That's it for today. We meet us tomorrow at same time, same place for some more exciting lectures about microscopy. Yeah. Then once we are done with the microscopy, we will take you to the even more exciting part of cell culture. We will see some virtual videos regarding that how cell passaging is done, how a normal cell culture life routine is, how to check your any contamination is happening. So we will touch each part and how to live a sterilized life while doing cell culture. That's the most important one. Yeah, you can say that. So Anjali, it's actually wherever you're gonna work in the lab, they will have the microscope that you will use. It's like the lab, when the lab is being set up, when the lab manager is buying the microscope, so they will buy according to the PI. Your PIs mean your principal investigator, your professor. So they will have the correct microscope for your use. Yeah, if you're having 4 or 5 different microscopes, so as we are discussing now, as per the application, you can choose. You can go for all microscopy actually. No one will stop you to use it. But at the end of the time is really crucial in the research. We can get better images, but at the end of the day, if you are in the lab, you're having a confocal microscope, that should be your goal to get your images. At the confocal microscope, you get really very nice images of any research that you're doing, and most of the high published applications, papers, publications, they're taking confocal microscopy as the most prominent one. I will also share some slides from my PhD. I was not so good in the microscopy. I had a very hard time to do good microscopy with antigens and antibodies, and but I was successful by 1 month at the end. So it's all depends upon your hand working. How good are you handling in the things? How good are you looking at the things in very, in detailed manner? How good are you patient? How good you can identify things when you having your eyes under your eyepiece and seeing the material under the microscope. So it's many factors that depends to have a better images, better results, um, to get a good results also. Yeah. So on that note, thank you very much. We will go for tomorrow another lecture for microscopy. Until then, take care, stay safe. Bye bye.